have identified the oldest samples of DNA ever found on Earth.
A cave in the Altai Mountains of Siberia, 2008. An archaeologist crouching in the sediment finds a fragment of bone, a finger bone, tiny, from a child, maybe 10 years old. The fragment is smaller than the tip of your thumb. They send it to the Max Planck Institute for Evolutionary Anthropology in Leipzig for DNA analysis. They expect Neanderthal remains. The Altai Mountains have Neanderthal deposits going back 40,000 years. This is routine. The result comes back in 2010. It is not Neanderthal. It is not anatomically modern human. It is not anything in the database. It is an entirely new species of human being. Everything we thought we knew about who was alive 50,000 years ago was wrong. The bone is from a species now called the Denisovans, named for the cave where the only physical evidence of their existence has been found. To date, the entire physical record of the Denisovans consists of 1 finger bone, 3 molar teeth, a partial jawbone found in Tibet, and fragments of bone from a second individual recovered in 2019. That is everything. A species of human being that shared the planet with us, that interbred with our ancestors, that left genetic material in the DNA of living people alive right now. And all we have found of them physically can fit in a small envelope. What the DNA told us is extraordinary. Denisovans split from Neanderthals approximately 400,000 years ago. They were as genetically distant from Neanderthals as Neanderthals were from us. And yet, all 3 groups were alive simultaneously in overlapping regions of Asia and Europe for tens of thousands of years. The Denisova cave alone contains evidence of all 3 species in different sediment layers, sometimes separated by as little as a few thousand years. In one extraordinary case, a fragment of bone from the cave belongs to an individual with a Neanderthal mother and a Denisovan father, a first-generation hybrid. Direct evidence of interbreeding between 2 species that we previously did not know existed simultaneously. This is not ancient history, this is in you. Modern Tibetan populations carry a specific genetic variant called EPAS1, a gene that regulates the body's response to low oxygen environments at high altitude. It allows Tibetans to live and work at elevations that cause altitude sickness in most people. It is one of the fastest evolving genes ever identified in the human genome. EPAS1 in Tibetans does not come from anatomically modern humans. It comes from the Denisovans. Approximately 40,000 to 50,000 years ago, modern humans expanding into Central Asia encountered and interbred with Denisovan populations. The offspring carried Denisovan DNA. Most of that DNA was gradually diluted out of the gene pool over generations. But EPAS1 was so functionally advantageous at high altitude that it was strongly selected for, and it persisted. The result is that a gene carried by a population of humans who are otherwise entirely unknown to us, a species we discovered from a single finger bone in 2010, is the reason the highest altitude human population on Earth can survive where others cannot. The Denisovans left almost no physical trace, but they are still alive in the bodies of roughly 6 million people. The Denisovans present a problem that geneticists and archaeologists are still working through. We know what they were genetically. We have their full genome, sequenced from the finger bone to a higher quality than the sequencing of most living populations. We know their population history, their migration patterns, their interbreeding events. We know that populations in Papua New Guinea and Aboriginal Australians carry up to 5% Denisovan DNA, the highest proportion of any living human population, suggesting extensive contact in Southeast Asia and the Pacific. What we do not know is what they looked like. The molar teeth recovered from Denisova cave are extraordinarily large, larger than Neanderthal molars, larger than modern human molars, closer to what you would expect from a much older hominin. The Xiahe mandible from Tibet, found at 3,280 meters altitude, shows a jaw structure that is robust, archaic, and unlike anything from anatomically modern humans. But we have no skull, no face, no artist's reconstruction based on physical evidence. Every image of a Denisovan you have ever seen is speculation. We know this species intimately at the genetic level. We cannot tell you what color their eyes were. Denisova Cave is arguably the most important archaeological site in the world for understanding human prehistory. The sediment layers preserve a record going back 300,000 years. Neanderthals occupied the cave. Denisovans occupied the cave. Anatomically modern humans occupied the cave. The evidence of all 3 species is present in different strata. In 2019, a fragment of long bone, designated Denisova 11, yielded DNA confirming the first-generation hybrid individual. The individual's mother was Neanderthal, with ancestry tracing to a Western Neanderthal population. The father was Denisovan. They met, reproduced, and their child died in a cave that 3 species of human were using across thousands of years as shelter. The cave also contains the oldest known jewelry made by any hominin in Asia: a carved bone pendant, a polished tooth pendant, a bone needle with an eye. Whether these were made by Denisovans, Neanderthals, or modern humans arriving later remains contested. The objects predate the arrival of modern humans in the region by tens of thousands of years. The non-modern hominins in that cave were making jewelry, using tools, and doing things we had assumed were unique to us. We assumed wrong. The Denisovans are almost certainly not the only species of human we don't yet know about. Homo floresiensis, the so-called hobbit species from the Indonesian island of Flores, was discovered in 2003. Adults stood roughly a meter tall. They used stone tools. They survived until at least 50,000 years ago, overlapping with modern humans. They do not match any known human species, and DNA extraction has so far been unsuccessful due to the degradation of bone in tropical climates. Homo luzonensis from the Philippines announced in 2019, a mosaic of features, some primitive, some derived, that doesn't fit any known human lineage. The Red Deer Cave people of southern China, alive until approximately 11,000 years ago. Archaic skull features that don't match any identified species. Fossil DNA analysis is still in its infancy. The cold, dry conditions of the Altai Mountains preserved the Denisovan finger bone well enough for sequencing. Warm, humid environments, which cover most of Southeast Asia, where the evidence suggests Denisovan populations were most widespread, degrade DNA within thousands of years. We are finding the species that happen to die in the right conditions. The others are gone without any trace we can currently read. Here is what the finger bone from Denisova Cave means for you. If you have any ancestry from East Asia, Southeast Asia, South Asia, the Pacific Islands, or Aboriginal Australia, you carry Denisovan DNA. The proportion is small, between 0.5% and 5%, depending on population. But it is real, it is functional, and some of it is doing specific things in your body right now. The EPAS1 variant in Tibetan populations is the clearest case. There are others. For the rest of the world, European and African populations carry essentially no Denisovan DNA. This is still your history. Modern humans did not arise in isolation. They arose in contact with other human species, interbred with them, absorbed their adaptations, and carried those adaptations forward. The human species as it exists today is not pure. It is a composite. You are not the end product of a single clean lineage. You are the survivor of a world where multiple kinds of human being were alive simultaneously, and most of them are now gone. We found one of them from a finger bone. We don't know how many we haven't found yet. The Denisovan discovery was not an isolated revision. It arrived at the end of a decade in which almost everything the scientific community had assumed about Neanderthals was being overturned. For most of the 20th century, Neanderthals were characterized as a dead end, a brutish, less intelligent human type that was outcompeted and replaced by behaviorally modern Homo sapiens arriving from Africa. The archaeological evidence was interpreted to support this. Neanderthal tools were simpler, their art was absent or rudimentary, their burial practices were less elaborate. The genetic evidence dismantled this characterization systematically. First, the interbreeding data. Non-African modern humans carry 1 to 4% Neanderthal DNA. This is not a trace. This is a documented, functional genetic contribution from a population of people who were supposed to be our competitors. They were not only our contemporaries, they were in some cases our relatives. Second, the cultural evidence, re-examined. The Châtelperronian tool industry in France and Spain, previously attributed to early modern humans, has been redated to Neanderthal occupation levels. The pigment use at Spanish sites at Cueva de los Aviones dates to 115,000 years ago, at least 20,000 years before modern humans arrived in Europe. Neanderthals were using pigment for decoration, ochre, manganese dioxide, at a time when our ancestors were still confined to Africa. Third, the cognitive evidence. Studies of Neanderthal hyoid bones, the small horseshoe-shaped bone associated with speech production, show a structure functionally indistinguishable from that of modern humans. Neanderthals could almost certainly produce the range of sounds required for complex speech. The assumption that they couldn't communicate at our level has no anatomical support. The story we told about Neanderthals, inferior, doomed, replaced, was a story we told about ourselves. We needed them to be lesser so that our dominance made sense. The DNA says something different. 50,000 years ago, the world had at least 4 species of human being alive simultaneously. Homo sapiens, expanding out of Africa into Europe, Asia, and eventually the Americas and Pacific. Neanderthals, present across Europe and Western Asia, in populations that had survived the previous glacial maximum and were adapted to cold environments in ways that modern humans were still learning. Denisovans, present across Central and Eastern Asia, with populations extending into Southeast Asia and possibly into the Pacific island chains. Homo floresiensis, on the island of Flores in Indonesia, a population that had been isolated long enough to evolve significant physiological differences from the mainland populations it had descended from. These were not separate worlds. They were overlapping ranges, contested resources, and in multiple documented cases, direct contact and interbreeding. The standard model of human prehistory, a single species moving across an empty world, filling ecological niches that nothing else occupied, is not accurate. The world that modern humans expanded into was already occupied by multiple human relatives who had been there for hundreds of thousands of years. The expansion of Homo sapiens was not a colonization of empty land. It was a displacement, possibly a conflict, and in some cases a merger. We carry the record of that merger in our DNA. The Tibetan breathing at altitude using a Denisovan gene. The immune variants that non-African populations carry, some of which trace to Neanderthal sequences, that provide specific resistance to pathogens that were circulating in Europe and Asia before modern humans arrived. The shade of your skin, the efficiency of your metabolism at cold temperatures, your specific distribution of fast twitch and slow twitch muscle fiber, none of these emerged in isolation. They emerged from contact with populations that had been adapting to their environments for hundreds of thousands of years before we got there. You are the outcome of 50,000 years of merging.